we're done with mitosis. We have to do the cytokinesis step. But the fact is, cytokinesis actually overlaps slightly with telophase. As telophase is starting to finish, cytokinesis is already beginning. As a matter of fact, on the diagram up there, you can see the cleavage furrows, which are a hallmark of cytokinesis, at least in animal cells they are already starting to occur during the telophase. And there we go, the two daughter cells. And you know, if these are rapidly dividing cells, like the zygote growing into a baby, each of those two daughter cells will then immediately go into interphase and begin the next round of the cell cycle. Okay, so any questions on that? Kind of a big recap of Wednesday's lecture from last week. Okay, I think I finished off last time showing you some photographs of cells going through the cell cycle. Let's look at those again. Here's a animal cell in the first part of prophase. You can see the chromosomes just starting to condense there. And this is a little bit later on in prophase. The chromosomes are fully condensed, and you can see the centrosome here and a centrosome there. You can't really see the two centrioles that make up the centrosome, but they're in there somewhere. And you can see some spindle fibers starting to grow out of the centrosome. And here's metaphase. The chromosomes are lined up nicely in the middle. You can see the centrosomes very clearly there. And then in anaphase, the duplicated chromosomes divide. One set of 46 unduplicated chromosomes going there, and one set of 46 unduplicated chromosomes going towards there. Telophase, they're separated even more. You can see the cleavage furrows starting to form. And cytokinesis, now you've got two daughter cells. And here's the same thing, but in plants. Prophase, metaphase, anaphase, and telophase. You can see the cell plate, which is that new cell wall that gets forming right there between the two daughter cells. Oh yeah, I think I showed you this last time. These are more plant cells. This is from an onion, I think. And you can look around there, you can see examples of pretty much all these phases we talked about. This one is pretty clearly in interphase because it's chromatin, not any condensed chromosomes. This one looks like it's in the middle of the anaphase, I guess. This one looks like it's just after telophase, so it's early cytokinesis. You can see the cell plate right there, and the two nuclei of the two daughter cells already going back into the chromatin state. Anaphase. This one maybe telophase right here. Where's a good? This is probably early prophase. You can see the chromosomes are pretty well condensed right there. Where's there a metaphase? Is there a metaphase? Oh, here's a nice metaphase right there. Everybody's lined up in the middle. Alrighty. Alright. Well, let's see now. Somewhere here. What's the next part? All right, so there's that diagram of the cell cycle again. So cells that are supposed to be dividing are in the cell cycle. They're alternating between interphase and mitosis and cytokinesis as they divide and divide and divide. And there's certainly some cells in your body that are constantly in the cell cycle. A good example are your skin cells. You're constantly flaking off skin cells from the outside of your skin, and those constantly have to be replaced. And so a little bit deeper than the surface of your skin is a layer of skin cells whose job it is to constantly divide and to replace the ones that fall off. Or another good example is the lining of your stomach. It manages to digest itself constantly at a slow rate, and so those cells have to constantly be in the cell cycle to replace themselves. Blood cells are another good example. there are some cells in your bone marrow that constantly divide to make new blood cells for you. But so some cells are constantly dividing, but there are other cells in your body that don't normally divide. And a good example of those are neurons, nerve cells, like your brain and your spinal cord are made out of. And those guys are not within the cell cycle. They're non-dividing cells. And so the cell cycle has things in it called checkpoints, which are stopping points for cells that aren't supposed to be dividing, or for cells that aren't ready to divide at a certain point. And in fact, there are 3 checkpoints within the cell cycle, 3 stopping points. There's one checkpoint right at the end of G1, in other words, right before the cell can enter S phase, there's a checkpoint. There's another checkpoint right at the end of G2. Think of that as like a stop sign for the cell that can stop it from entering prophase. And there's another checkpoint right here, right at the end of metaphase. Think of that as a stop sign that can stop the cell from entering anaphase. And, you can continue making an analogy with stop signs and roadways. When you drive your car down the road, there are certain regions where you're supposed to stop your car if it's appropriate, and other parts of the road where you're not supposed to stop your car. And anyway, these are the agreed upon stopping points if the cell needs to stop itself within the cell cycle. Let's see now. I want to mention at a molecular level why the cells stop at these particular checkpoints. And the answer is that the enzymes that carry out the phase after each checkpoint just aren't turned on yet, just aren't activated yet. And to illustrate what I mean by that, let's consider S phase for a moment of interphase. It's kind of a review question. What goes on in S phase? Can somebody just briefly describe what activity occurs during S phase? The what? The chromosomes duplicate or replicate. That's right. You go from each chromosome in the cell goes from being an unduplicated chromosome to being a duplicated chromosome, the 2 sister chromatids on it. And here's another review question. Who can name some of the enzymes that carry out that process? I gave you a whole list of them when we talked about like the replication fork and things like that. What was it? DNA polymerase. There's a number though. DNA polymerase 3. Good. And before that can operate, you need something to make a little piece of RNA to start it off. What's that called? Primase. Yeah, there's an enzyme called primase that makes those little RNA primers to get DNA polymerase 3 started. The primers eventually get turned to DNA. What's the name of the enzyme that joins them together? Ligase. Yeah, there's an enzyme called ligase. And I'll just say etc. because there's a bunch of other enzymes involved also. There's an enzyme that converts the RNA primers into DNA primers before ligase can operate. There are enzymes that uncoil the DNA at the replication forks. I don't know exactly how many enzymes are involved in duplicating the chromosomes, but at least dozens. It wouldn't surprise me if there were hundreds of enzymes involved in going from there to there. And so the point I'm making is the reason why the cell stops at G1 is those enzymes that carry out S phase, the DNA polymerase 3 and the primase and the ligase and all the other ones, just aren't activated yet. They're just not in their activated state yet. And so if we activate those enzymes, then we move past the checkpoint. Then that's what puts us into S phase, is activating those particular enzymes. And the same with the other checkpoints. Why does the cell stop here in G2 without entering prophase? Well, because the enzymes of prophase just aren't activated yet. What happens in prophase? The chromosomes condense, so the enzymes that condense the chromosomes just aren't activated yet when you're in G2. And the enzymes that break down the nucleus aren't activated yet, so that's why the cell stops there at that checkpoint. And what else? So the enzymes that make the spindle apparatus in prophase just aren't activated yet. And so that's why the cell is stopping and it's not able to carry out prophase yet because the enzymes aren't activated yet. And same here, what happens in anaphase, the centromeres divide and the chromosomes move apart from each other. The enzymes that carry out that process are just not activated yet in metaphase, and so that's why the cell will stop there until those enzymes are activated. So it's all about carrying out the enzymes that carry out the phase after the checkpoint not being activated yet. And when I was writing up these lecture notes, I looked through a whole bunch of biology textbooks to say, well, is there a general term, to see if there was a general term for the names of the enzymes that carry out each phase, and there doesn't seem to be. And so I kind of made up my own. I called them mitosis phase enzymes. Now I can't see what I wrote that down. Well, I thought I had that phrase on here, but I call those mitosis phase enzymes. For each mitosis phase, the enzymes that actually carry out its functions are the mitosis phase enzymes. All right. So what I want to do is look at these checkpoints in a little bit more molecular detail and see exactly what's going on at the checkpoint, what exactly makes the cell stop, and how does the cell get past that particular checkpoint when it's appropriate for the cell to move past that checkpoint. And let's start off with the G1 checkpoint. This is probably the most important checkpoint of the cell cycle. It has a couple of special names. It's sometimes called the restriction point. And the reason why I say it's the most important of the three is it's the normal stopping point for cells in the body. There's a lot of different varieties of cells in your body, skin cells and stomach cells and bone cells and cartilage cells and blood cells. And some of those cells are constantly actively dividing, but many of them are not dividing at all. And the cells in your body that are not dividing, that's where they park themselves at, is at this G1 checkpoint. And so I guess that's why they call it the restriction point, because most cells are restricted to just being at that particular spot in the cell cycle and not moving past it. It's sometimes also called the GO phase for cells. I guess O means zero, so it's not G1, it's not G2, it's not interphase, it's not cytokinesis or mitosis or anything. So some people call it the GO phase, and other people just say the cell has left the cell cycle. Elvis has left the building. The cell just not actively divided, it's in GO phase. Now, let's see. Well, as I've been going on about, there's many different cell types in the body, but you can generalize about certain cells being in the cell cycle and some not in the cell cycle. Some cells are constantly dividing. And I guess I mentioned some of these skin cells are constantly dividing cells. I wonder if I have some pictures here. Well, skin cells are a good example. Bone marrow cells, the ones that make your blood cells, are constantly dividing. But those are good examples. Whereas other cells are not dividing. So these ones you could say are in GO phase, or they're at the restriction point, or they've left the cell cycle. Any of those phrases mean the same thing. But focusing on this group of the non-dividing cells in your body, you could break those down into two further categories. Some of them are permanently non-dividing. And a good example of those are nerve cells. And another good example are muscle cells. How do you think about this? This is consistent with our experience of injuries. You know that if people damage their nerve tissue, maybe they're in an accident and tear the nerve tissue in their spinal cord, those people will be paralyzed for the rest of their life because those nerve cells will never regenerate, will never go through mitosis and replace themselves because they can't. Just by definition, nerve cells are just permanently stuck in GO and they can never re-enter the cell cycle. And the same with muscle cells. If you lose a chunk of muscle from your body somehow, maybe in an accident or something, the other muscle cells will never regrow and go through mitosis and replace those missing muscle cells. However, there are other non-dividing cells in your body that can get back into the cell cycle if they have to. Good examples of those are a type of cell called a fibroblast. Those are scar-making cells.